So the action potential that have propagated along the axon now have reached to the synapse, which is the gap between one neuron to another neuron. Okay, there are two types of synapse. The first one is electrical synapse. So this consists of a gap junctions that allow ions and small molecules to flow directly from one neuron to another neuron. Okay, presence of this gap junction. So transmissions across this synapse is very rapid, and this is common in heart and the brain because the nerve signal need to maintain steady and rhythmic muscle contraction. The second one is a chemical synapse. There is a gap, there is a narrow gap that we call as a synaptic cleft that separate the sending neuron, or we call as a presynaptic neuron, from the receiving neuron, or a postsynaptic neuron. So this is receiving, and this is the axon that send the impulse. And the end of a presynaptic neuron here is swollen and filled with a synaptic vesicle that are packed with the neurotransmitter. So as the action potential reach to this synaptic terminal, it will then cause depolarizations of this presynaptic membrane. So this neuron here is a presynaptic neuron, and this is a postsynaptic neuron. And this is the synapse, which is the gap between one neuron to the next. Okay, so I just shown here this diagram here indicate by these two bulbs of axon terminal. So now action potential reach to this terminal, okay, and there are vesicle, synaptic vesicle that containing neurotransmitter at this synaptic terminal. So action potential will depolarize the presynaptic membrane. Okay, and depolarizations open voltage gated calcium channel. So there are voltage gated calcium channel that cause and trigger the influx of calcium into the neuron. Okay, and as these calcium concentrations inside the presynaptic terminal increase, it will then trigger this synaptic vesicle to fuse with the plasma membrane. Okay, and as these fuse with the plasma membrane, it will then release the neurotransmitter by exocytosis. So neurotransmitter will then release to the synaptic cleft. So the narrow gap here we call as a synaptic cleft. Okay, so release of a neurotransmitter to the synaptic cleft via exocytosis. So what happened next? The neurotransmitter here will then bind to this ligand gated channel. Okay, ligand gated ion channel in the postsynaptic membrane. Okay, this is the postsynaptic membrane. So this neurotransmitter will diffuse and bind to this receptor. Okay, so this receptor will then cause the opening of the receptor of the channel, ion channel here, and cause diffusions of ion across the membrane. Okay, so this is how neuron transmit impulse from one neuron to the next of a neuron, so that the impulse can be transmitted to the next. So now when neurotransmitter bind to the ligand gated ion channel on the postsynaptic neuron, it can trigger either excitatory postsynaptic potential or EPSP, or can cause inhibitory postsynaptic potentials or IPSP. So remember, this is a potential on the postsynaptic neuron. So excitatory postsynaptic potential, or EPSP here, when the ligand-gated ion channels are permeable to both sodium and potassium. So it will cause sodium to move into the cells and cause potassium to leave out of the cells. So this will create positive charge inside these cells and result in depolarizations of a membrane, okay, that bring membrane potential toward the threshold and then generate action potential in this next neuron. Okay, so this is when it activates the next neuron. While inhibitory postsynaptic potentials, or IPSP, when the ligand-gated ion channel permeable to potassium ion and also permeable to chloride ion. So chloride will move into the cells and that will result in hyperpolarizations of a membrane, and this will bring membrane potential away from the threshold. So it will inhibit the next neuron, so it will not generate action potential. So after a response is triggered and these postsynaptic neurons have been activated, the chemical synapse needs to return to its resting state. Okay, so neurotransmitter here, when it's bound to this ligand-gated channel, it cannot continue binding in here. So there are two mechanisms to terminate this neurotransmitter signaling. One by enzymatic breakdown of neurotransmitter in the synaptic cleft, which means there are enzymes that will break down this neurotransmitter and then they are going to be recycled back to the presynaptic neuron and packed again into the vesicle. The second mechanism is reuptake of neurotransmitter by the presynaptic neuron. So this neurotransmitter unbound again with this receptor and then uptake, reuptake again into the presynaptic neuron. So clearing neurotransmitter from synaptic cleft is very important. So blocking of this process can have severe effect. So example here like nerve gas serine that inhibit enzyme that break down neurotransmitter, which is nerve serine gas here inhibit acetylcholinesterase. Okay, so acetylcholinesterase is supposed to break down acetylcholine, which is one of the neurotransmitter. By inhibit this enzyme, mean enzyme here cannot break down the neurotransmitter. So neurotransmitter keep binding on this receptor and keep activating the postsynaptic neuron. Okay, it will trigger paralysis and eventually death. Neurotransmitters are referred as body's chemical messenger. So you have learned previously on how nervous system use this neurotransmitter to transmit messages between neuron and also from neuron to a muscle. So here are a few major neurotransmitters that have been grouped into their classes. So the first one we have here is acetylcholine. This is the first neurotransmitter to be discovered and the only neurotransmitter in its class. So acetylcholine can be found in both central and peripheral nervous system and play a role in muscle movement as well as in memory and learning. So acetylcholine are neurotransmitter that is released in the neuromuscular junction, which is junction between neurons and muscles. We call it the neuromuscular junction. So acetylcholine are the neurotransmitter that are responsible for your muscle movement and allow you to move around your hands and your leg. The second one, which is in a group of amino acid, is a glutamate. Okay, so this is the main excitatory neurotransmitter in CNS. Excitatory. Okay, because it's going to stimulate or activate postsynaptic neuron. postsynaptic and presynaptic. So remember, the generations of postsynaptic potentials can be IPSP or EPSP. So once glutamate released as a neurotransmitter in the synaptic cleft, so this is glutamate release, bind to this receptor, and then it will cause depolarization to the postsynaptic neuron, that it causes the neuron to transmit the action potential. Okay, so that is meant by excitatory neurotransmitter. Okay, so which means it will generate EPSP on the postsynaptic neuron. So compared to its derivative like GABA and glycine, so GABA stands for gamma aminobutyric acid. So these two are inhibitory neurotransmitter. Inhibitory. So inhibitory means it will not stimulate the postsynaptic neuron, however, it will inhibit. Okay, so let's say here GABA being released as a neurotransmitter on synaptic cleft and then bind to this receptor on the postsynaptic membrane, then cause hyperpolarizations, okay, on the next neuron, or generate, not EP but IPSP, inhibitory postsynaptic potentials. So how GABA can cause IPSP? Because it increases permeability to chloride ion. So as it binds to the receptor, it will cause chloride to move into the cells and hyperpolarize the postsynaptic neuron. Okay, so same goes to the glycine, both are inhibitory. And the next are group of biogenic amines, neurotransmitter, which include norepinephrine. So norepinephrine involved in fight or flight response. So dopamine, this is a neurotransmitter that responsible for brain's reward system. Okay, that make you rush for a joy when you accomplish a goal or succeed at a task. While serotonin, this is very important in regulating mood, anxiety, sleep, and appetite. And the next is a group of neuropeptide, which is a diverse group. And one of the examples here is an endorphin. So endorphin is neurotransmitter that inhibit transmissions of pain signal and promote feelings of euphoria. And the last group that we have here is a gaseous neurotransmitter. As an example, nitric oxide. So nitric oxide play a role in affecting the smooth muscles, relaxing the smooth muscle to allow blood vessels to dilate or increase the blood flow to a certain areas of the body. In advanced technology, the introductions of mind-controlled robotic prosthetic allow patients with amputations, traumatic injuries, or those who were born without a limb to utilize a complete and full range of motion. A prosthesis is an artificial device that replaces missing body parts. It consists of several components to integrate with the body function. So the biosensor receives signals from nervous system, meanwhile the mechanical sensor process information affecting the device such as the positions of the limb and the applied force. So following amputations, the nerve endings remain. So these nerve endings that initially supply the neurons to the muscles now remain. Then these nerve rerouted to a healthy muscles, reroute again to a healthy muscles, and this is what we call as a targeted muscles reinnervation. So this targeted muscle reinnervation is a surgical procedures that reassign the nerve that once controlled the arm and the hand, so now reroute to the healthy muscles. Okay, so now when patients start to think about moving arms, the muscles will contract. And the electrode here, so the electrodes that attach to muscle respond to the thought, because these electrodes are going to pick up the activity of a neuron. So patients start to think about moving their arms, and that trigger transmissions of impulse along the neuron and pick up by the electrode. So the electrode here will detect the movement and send the signal to a processor. So here the computer processes the signal from electrode, and then the motor in the arm provide the movement. So the advanced robotic prostheses are equipped with more than 100 sensors that can detect stimuli such as pressure and temperature. Okay, so not only the thought from the brain that sends signal to this bionic arm, but they can also detect the external stimuli such as pressure and the temperature and send to the brain. So we have come to the last topic in this chapter on comparison between endocrine and nervous system. So we know that both of these systems involve in internal communications and regulation. But what are the difference between these two? So we are going to look at the difference between endocrine and nervous system. So first, type of signal. Endocrine release a chemical signal while nervous release an electrical signal. In terms of speed, traveling of speed for the signal, it must lower in endocrine because the chemical signal transported by blood, but it's really fast. The signal travel really fast in the nervous system, especially due to the saltatory conduction, where the impulse can be transmitted within millisecond to second. Even though the message can arise really fast to target site in the nervous system, the durations of effect is very short and prompted for the nervous, but it's a long-lasting effect on the endocrine system. Speed of response, endocrine provides slower response and nervous provide rapid response. And the last one, in terms of accuracy of a message, is more diffuse in endocrine because the signal is spread out, but it's more precise on the nervous system because the neuron that is released transmit directly to the target site. So that's the end of chapter 5. I hope everyone can understand on this topic, and thank you.